There's a lot of you here, which is amazing. So just give me a thumbs up because obviously yesterday there was a lot of issues with the sound quality. I hope you can hear me. Okay, I assume you guys can hear me, thank you. So let's get started. So how this live stream is going to work is, you may have seen already, we're going to go through a lot of exam questions. And we're going to start, in this live stream, we're going to go through the first half of the topics, which you can see on screen now on the left-hand side. And then on the night before the exam, we're going to go over the second half of the topics. And how we're going to go over the topics is we're going to go over exam questions to help revise each part of the topic. Now, if anyone has any questions, you can just put your questions in the chat and I'll stop hopefully and try and answer your questions. But I want to get through as many questions as possible. So let's get started. I recommend you trying to answer the questions alongside me doing it as well. Okay, so starting off with eukaryotes and prokaryotes. So the first question, recall and describe the function of a typical structure in a prokaryotic cell. So in terms of doing the exam questions, you can try and type it into the chat, or you can maybe just say it to yourself and then check as we're going with the answers. So let's think of how to answer the first question. So describing the function of a typical structure in a prokaryotic cell. One sec, I'll just check that the stream is working on all fronts, because I am streaming on different platforms as well. This is for combined higher and triple, so mainly triple, and if you're doing combined higher, this will be beneficial to you also. Okay, so I can see people have already started answering, so let's get started with answering these questions. So cytoplasm, let's think of the couple of things in prokaryotic cells. So you've got cytoplasm, you've got cell membrane, you've got cell wall, bacterial DNA, plasmid DNA, etc. So these are the different parts of the prokaryotic cell, and then you've got to obviously explain what those different functions are. Now, I don't want to show you the answers just yet because I want you to think about possible answers. So yeah, cytoplasm, what is the function of it? This is where most chemical processes occur. Cell membrane, what is the function of it? It controls the movement into and out of cells. Cell wall strengthens the cell. Bacterial DNA, because you can talk about bacterial DNA or you can talk about plasmid DNA. Both are two different parts of prokaryotic DNA. Okay, bacterial DNA controls the function and synthesizes proteins. Plasmids are rings of DNA used as a vector in genetic modification. This would also be beneficial for foundation, but I don't want to confuse you guys. So it's mainly for combined higher and triple, and it's for the AQA exam board, okay? Let's look at the next question. Give 3 differences between a prokaryotic and a eukaryotic cell. So prokaryotic cells are smaller than eukaryotic cells. Prokaryotic cells contain genetic material that is free, it's freely suspended in the cytoplasm. Whereas eukaryotic cells, the genetic material is obviously enclosed in a nucleus. Prokaryotic cells have plasmids. So you don't have to go into depth because you only need to think about 3 differences. By the way, the questions that I'm giving you, these are also questions that most students have got wrong in previous papers. So I've purposefully selected them. Okay, really good examples and answers in the chat. And don't worry, I'm going to show you the answers in a second. Let's just look at the last question. Give 2 similarities between prokaryotic and eukaryotic cells. So you could say they both have cytoplasm, they both have cell membranes, or they both contain genetic material. So those are the answers. You can screenshot it or come back to the live stream if you want to take note of any of those answers. But let's now move on to the next question. Okay, explain why mitochondria in cells is important. So this is animal and plant stem cells. So this is a question where a lot of students get wrong, and for a specific reason. So firstly, think about what you would say. Now, you need to say it in a three-step process. Cells require energy to function, or you can say something like, for work to be done, cells require energy. You can then say mitochondria is the site of aerobic respiration. Don't just say respiration, aerobic respiration, well done. And then you want to say energy is released in respiration. The most common error with the question about why mitochondria in cells is important is because people say energy is made. Yeah, exactly. Energy is released. Energy is not made because of the law of conservation of energy, which you learn in physics. Energy cannot be created or destroyed. Hence, we know that energy is released. Perfect. Okay, moving on to the next question. Ribosomes synthesize proteins. Explain what this means. Okay, so this is an outlier question. It's a bit more difficult, and that's why obviously I brought it on the screen. So all you would have to say is synthesize means to produce from amino acids. So ribosomes are the site of protein production. Okay, now if you need to go over that, don't worry, just pause when I show you the answers. The next one, what is the function of a nucleus? Now, this is one marker, but the function of a nucleus often is a two marker because there's two key functions of a nucleus. The first one is to control the activities of a cell. The second one is that it just contains genetic material. Okay, so the two functions of nucleus: controls the activities of the cell and contains genetic material. Okay, the next one. Name one type of cell that does not contain a nucleus. So here it's quite open-ended. You can give any answer from red blood cells, bacteria, prokaryote, xylem cells, etc. Okay, so pause if you want to see the answers for this one. If not, let's move to the next question sheet. Okay, compare sperm cells and nerve cells. So this is 3 marks. So firstly, you need to talk about the function of the sperm and nerve cell, and then you want to talk about the different adaptations. Okay? So, sperm cells. The function of a sperm cell is to fertilize an egg. That's all you have to say. Function of a nerve cell is to send electrical impulses throughout the body or to the brain or whatever along the spinal cord. Now, adaptations, we know sperm cells, they're streamlined, they have a long tail, or often known as a flagella. They also have enzymes which are in the head of the sperm, which is called the chromosome. And these enzymes help to break the egg for fertilization. As people have said in the comments, yes, sperm cells contain a lot of mitochondria. Well done, guys, for interacting in the chat as well. You're doing really well. Okay, yes, sperms did win the race. So nerve cells send electrical impulses to the brain. They have a very long axon, which is like a long body. And because of that, that allows them to transmit electrical impulses really, really quickly. They contain dendrites, which are branched out, and they act as the insulating sheath. Okay, and those are the things you can say. You can also mention some other things such as myelin as well. Next, what does differentiation mean? Okay, so here's a 2-step answer. Differentiation. Differentiation is how a cell changes over time. And then your second point is to become specialized for a specific function. Okay, compare animal and plant stem cells. So, animal stem cells differentiate early in their life, and that's why we call them embryonic stem cells. Therefore, animal stem cells are mainly used for replacement or repair. Dendrites is not necessarily a triple concept, it's just something you learn with nerve cells. The reason why I spoke about, I gave a question about nerve cells is because most people actually forget the adaptations of nerve cells. So that's why I mentioned that. Okay, Yes, plant cells, exactly. They can differentiate throughout their life, whereas animal stem cells differentiate early in their life. And therefore, animal stem cells are used mainly for replacement or repair, whereas plant stem cells are grouped together in a structure called the meristem. Exactly, they're found in the embryo and bone marrow. That's a really good answer as well. Exactly, so the meristem. So sometimes you call that meristematic tissue. Okay, and moving on to the 4 marker at the bottom now. Explain the advantages and disadvantages of using plant stem cells. So, thinking of advantages, rare species can be cloned. Crops with useful adaptations can also be cloned. Disadvantages include if you actually start using stem cells to clone a certain species of plants, what happens is it could actually start competing with other plants and put other plants in danger. Okay. Another disadvantage could be that the cloned plants, because they have the same genetic makeup, they're more susceptible to diseases because they have identical genome. Exactly, they could be wiped out by a pathogen. So those are the answers. Let's now move on to the next topic, microscopy. We're going through this really at a good pace, so if I am going too fast, do tell me and I can slow down. Okay, moving on to microscopy. Now you can see I've just put the required practical, and this is 6 marker. What I just want you to do is kind of blurt out everything you can remember about the microscope required practical. This is important, and I want you to talk about things such as possible errors, because when you're actually applying your knowledge in the exam, you need to have a certain way of saying things. Now, what they could do in the exam is give you a slightly different version of the practical, so it seems unfamiliar, but the method is going to be the same. So going through the method, firstly, let's look at the equipment required for the microscope practical. Obviously, a light microscope, your glass slide, your cover slip, scalpel, your iodine, and you can talk about some further things as well, more specific things if you're cutting the onion or whatever cells you're using. Perfect. So the first step is preparing the slide, okay? So you're going to cut a section of your onion. You're going to peel the epidermis, which is that thin layer on the onion. Okay, you're going to peel it because you want to get the thinnest layer so you can see one single layer of cells. So once you've got this epidermis, you're going to place it on your glass slide, and you have to be very careful. Yeah, well done, guys, in the chat. I'm just reading your answers. They're very good. Okay, so you're going to place it on the slide and you're trying not to trap any air bubbles. Okay, so you can use forceps for that. Then what you're going to do is you're going to stain your specimen using iodine. Now, they might ask you, why are you using iodine to stain it? Well, you want to say to highlight the subcellular structures, okay? Next, you're then going to soak any excess iodine, because it may have dripped elsewhere, onto a paper towel. Because you don't want there to be air bubbles when you're putting the cover slip down, because that's obviously going to fragment your view through the microscope. You're then going to put your glass slide onto the microscope stage, and then move it up using the coarse focusing lens, and then adjust your view using the fine focusing lens, and then you'll be able to see it. Okay, and then you can talk more about that in detail if you're asked about that. Okay, moving on to the next question. Suggest one reason why the student couldn't see clearly through the microscope. So since they're talking about the seeing through the microscope, rather than why it's not working, perhaps why the slide isn't clear, or you wouldn't say any air bubbles because this is an error with the student looking through the microscope. So here you could say perhaps there was a dirty objective lens, or you haven't focused the microscope properly. Okay, now moving on to the next one. Compare the light and electron microscopes, giving advantages and disadvantages. Okay, so light microscopes, this is quite easy to say. So light microscopes, they're small, they're portable, they have a low cost, and they're relatively easy to use. Electron microscopes, they have a higher resolution, higher magnification. Perhaps they're more expensive, they're not portable, etc. And finally, give two advantages. Oh no. Give two advantages of using an electron microscope instead of a light microscope. So this is similar to what we've just spoken about, how electron microscopes have a higher magnification, higher resolution. And I've also included on the slide here, now I'm showing you the answers, a bit about how the light versus electron microscope works, just in case you were curious about what is actually the difference. So light microscopes use a beam of light to look at the cells, whereas electron microscopes use a beam of electrons, which have a very high wavelength. Okay, and it's actually a higher wavelength than light. So it's all talking about speed of light. In this video, we will be covering half of the biology content. The other half of the biology content will be covered the night before the exam, and this is for paper 1 AQA. Okay, let's now move on to the next topic, which is cell division and mitosis. You're doing really well, guys. Well done. Explain why mitosis is important for multicellular organisms, okay? So why do we need mitosis to happen? Now, a lot of people forget to actually learn this, so key things to remember. Mitosis repairs cells. It's really important because our skin cells are made via mitosis. So if we didn't have mitosis, every time we had dead skin cells, there wouldn't be anything to replace it. So it's really important to replace cells like that and repair cells as well. So if we were to hurt our arm and it would start bleeding, obviously, then you need to recover that blood, and blood is one of the cells that are produced via mitosis. So again, repair and replacement. And obviously that's a 3 marker, so you can talk more about how mitosis works. So mitosis is how cells are formed, identical cells. So there's 2 identical daughter cells, which are clones of the original cell. Okay, perfect. Now moving on to the next one. Describe what happens at each stage of mitosis. So thinking about the different stages. Stage 1 for mitosis, this is the longest stage. It's where everything is being replicated, all of the subcellular structures, including the DNA. Okay, so you could give some examples, say, for example, mitochondria that replicates itself. So there's double the number that there previously was. The second stage is slightly quicker. So what happens is one set of chromosomes is pulled to either end of the cells. So exactly, they're moving to opposite ends. And then the final stage is after, because the chromosomes have already separated and other things have already separated, mitosis, all of the subcellular structures. So the final stage is the cytoplasm and the cell membrane divide to form 2 identical daughter cells. Okay, and the last question for this topic, describe how mitosis produces 2 genetically identical cells. So again, this is a very similar answer, so I'll just show you. It's because this question was asked twice over the course of the previous years, and I wanted to show you 2 different examples of how you could answer that question. Okay, thank you guys for staying on live stream. I hope you're finding it useful. Let's now move on to the next topic, diffusion. How would you recommend splitting revision time for subjects? I would suggest just focusing on the exams for the day, during that day and during the night before. And then on the weekend, kind of split your time into the exams for the next week. Don't focus on exams that are not in that next week. That's what I did at least, and that's how it worked for me. Okay, moving on to the next one. What is meant by the term diffusion? Okay, this is a 2 marker. So you want to say the net movement of particles from an area of high concentration moving to an area of low concentration, going down the concentration gradient. Okay. In terms of whether chromosomes are subcellular structures, I think I misspoke there. So I meant that the nucleus is a subcellular structure and chromosomes are found within the nucleus. So well done for correcting me, guys. Well done. Okay. Next, explain the importance of maintaining a steep concentration gradient. This is a really interesting question, and a lot of students struggled to answer this when they saw the question. So, really, the answer is the greater the difference, the faster the rate of diffusion. And with the same concentration, no diffusion will take place. Okay? So, what they're effectively telling you is, firstly, what is causing what affects the rate of diffusion? Well, it's the concentration gradient, how different the concentrations are of the two areas. Okay. You want to say down the concentration gradient to be more specific. Yes, exactly. Well done, guys. And the next one. Describe the adaptations for transport in cells, looking at plants and animals. So obviously I've slightly adapted this question because I wanted you guys to be able to revise all of the different subcellular structures, whether it's in the small intestine, lungs, the plant root cells, the gills, etc. So I'll show you the answers now and we can go through them. So the small intestine, you've got the villi, which are the small finger-like projections. They increase the surface area. You've got good blood supply, lots of capillary network, and you've got a thin membrane. Lungs, oh yeah, xylem and phloem is also a good answer as well. Lungs, you've got the alveoli, which increase the surface area, good capillary network, thin for rapid transport, and elastic walls so that it can expand and contract. Yeah, as in active transport, so I think this was talking about any transport of gases, active transport, diffusion, osmosis, etc. Okay, gills. A lot of people struggle to understand how the gills work. So the gills are very thin. You can view them as like bits of skin that the fish have. Now, what happens is as the water goes through, the gills absorb the oxygen from the water via osmosis and diffusion. It's mainly diffusion. And you don't need to necessarily know about gills, but just know it's an example of an adaptation. Okay. And the lamellae are just the small projections on the gills. So they help to increase the surface area. Okay, let's now move on to the next set of questions. So these questions are about osmosis. So again, there's a required practical. So let's think about all of the points relating to that required practical. Okay, so yeah, gills is like bronchi outside. Thank you, Sophie, for the very nice comment. I hope you guys are finding this useful. Let's continue. So, looking at the required practical for osmosis, what are the key things here? Well, I wanted to introduce you to the term cork borer, which is kind of that metal cylinder which you use to cut into things. You may have done it in food tech. You may have used it to core your apples. It might be called a corer. So you're going to use that to cut potato cylinders. Now, a couple of years ago, instead of using potatoes in the paper for the required practical, they actually changed the vegetable to carrots or something. So just remember that they might change the actual vegetable if they want to confuse you, but make sure that you're using the same method. And yes, I can read the chat. So yeah, exactly. Cut the potato cylinders to the same diameter. You want it to be the same diameter because we want to keep that as the key variable we're not changing. Trim the cylinders to all of the same length because again, you don't want that to change. You want them to each be the same size, both in diameter and in length. Does it matter if I do Edexcel? This is more tailored towards AQA, but I know that there's a lot of overlap between Edexcel. However, obviously we're going through exam questions for AQA, so I don't want to confuse you in any way. Okay, you're then going to measure and record the length and mass of each potato cylinder. You're going to measure 10 centimeters cubed of the 3 different solutions of water. So they're all going to have different amounts of salt in each of the solutions. Okay, yeah, they're control variables. the independent variable is the solutions. So then you're going to add each potato cylinder into each of the 3 test tubes. You're then going to leave it overnight. Now, probably when you did this experiment in class, you didn't leave it overnight, you left it for maybe like 10 minutes. There wouldn't have been that much change, but typically if you leave it overnight, you're going to see a bigger change in mass. You're then going to remove each of the potatoes, and you're carefully going to pat them dry with a paper towel. Okay, and you don't want to squish them with your paper towel because you don't want the water that is just absorbed to leak out. So you need to be very careful with that. They may in fact ask you about that. Okay, and then you're going to remeasure the length and the mass, and then calculate the percentage change. Yeah, removing excess moisture, etc. Okay, now let's look at the next one. Explain how the solution could determine the concentration of the solution inside the egg. Now, this may be confusing at first, but what they're doing is an example of what I was talking about, how they've just switched it from talking about osmosis in terms of an egg. So again, you want to use the same idea, but apply that method to the egg practical. So here is the answers if you want to review them. Okay, and a key thing here, which I also want to tell you guys, as it's almost a secret tip, you plot it on the graph, and then what you do is you end up with a graph that it's hard to tell you, but it's a graph that has maybe I'll draw it out for you 1 second. That might be easier. Or I'll find it. And why I'm telling you about this graph is because there's a key point on the graph, which actually they may ask you about what does it represent. And then you want to talk about what that represents. Okay, let me see if I can share my screen. Yeah, exactly. Well done, Jude. Where 0% change, it's the initial mass of the egg. Let me share the screen. So you can see on this BBC Bite-sized website here, hopefully you can see the screen. No, you can't. So here you can see that where it hits the x-axis, that is the actual normal concentration of whether it's the egg or the potato. So that is where it's normal because there's no percentage change, which means that it's not undergoing any osmosis. Okay, and that's what your table of results would look like. Okay, so I hope that helped. Yeah, well done, Jude. Okay, let's now move on to the next topic, which is active transport. There's a whopping 1 question on here, so it's not been asked too many times. Explain how nitrate ions are transported into the root hair cell. So again, just explain the process of active transport. You need to talk about how it's not a passive process, it's an active process. So it requires energy to move the nitrate ions from lower concentration in the soil to a higher concentration in the roots. Okay, let's now move on to the next topic, which is principles of organization. Now, you may have initially been confused about what this means, but it's just talking about the different stages of organization, cells to genes to DNA to chromosomes. And reading this, I've just realized this is actually paper 2, so don't worry too much about this. Here's the answer if you want to look at it, but it's not necessary again for paper 1, it's for paper 2. Okay, now moving on to the next topic, the digestive system. So we're going on to the second kind of big topic in biology. The RS revision stream will be on the night before the RS exam. So I actually have a timetable, if you guys have seen, of all of the live streams. If you go onto my website, it should be there, or if you scroll down through the community on YouTube, you'll probably see a picture of it there. Okay, so now moving on to the digestive system. Explain the enzymes needed in digestion. Okay, so that's a weird way of phrasing it. Just explain the enzymes, but you just want to say enzymes are biological catalysts, and thank you, Wanya. The biological catalysts, and what they do is they cause the breakdown of large insoluble molecules into small soluble molecules, i.e., digestion, because you're not going to be able to digest big chunks of food. You need to break them down into small parts. Okay, and yep, you guys have already started naming them. That's really good as well. The mark scheme didn't say you had to name them, but it's still well worth doing that as well. That can be absorbed by the blood. Also, a tip for you guys, when you're answering the science questions, whether it's biology, chemistry, or physics, I would recommend you output everything you can remember about the topic, because the mark schemes are often super specific sometimes, especially if it's a difficult year because everyone is really revised well. So then if people have done well, they're going to make the mark scheme really annoying. So what you need to do is you need to bullet point. You can definitely use bullet points, at least in the AQA. Bullet point everything you can remember about a topic and then move on. Okay, enough of that. Moving on to the next topic. Question. Explain how the release of bile into the small intestine releases the rate of fat breakdown. Now, the number one mistake for this was people thinking it is an enzyme, and no, it isn't an enzyme. So, it neutralizes the acid. That's one function of it. Another function is that it emulsifies fats, so converts it into smaller droplets. Well done. I think someone in the chat said that as well. So this helps to increase with the surface area to volume ratio, and the previous one helps to maintain the optimum pH levels for lipase enzymes to work. Okie dokie. Yeah, exactly. If you're talking about RS quotes, I actually did make a document which has all the quotes you need. You'll find that on my website, and I'll release the Christianity and Islam one as well. Okay, explain why protease works more effectively in the stomach. Okay, so why does protease work more effectively in the stomach? Here you want to talk about the specific conditions that the stomach has, and you're talking about how that affects the rate of enzyme work. So the stomach is obviously acidic because of the hydrochloric acid. This means that there's overall a low pH, so therefore the enzyme, that must be the optimum conditions for protease to work. Okay, those are the answers. Okay, now moving on to the next topic, which is food tests. When I was doing GCSE Biology, this was my least favorite topic, but I know some people are quite good at this. Okay, so there's a required practical, I believe, about the food test, 6 marker. You don't need to know the actual pH, but it's obviously going to be acidic, so it won't be 7. It'll be like less than 7 for the stomach. Okay, pepsin, yeah, that's a good use of that. Okay, so let's look at the food tests. Again, we're going to blur everything we can remember. So firstly, a lot of the steps that people forget is actually preparing the food. Like, to do the food test, people often just say, for the protein, it's this test, it turns this color to this color. But remember, we actually need to break the food down in order to do the test. So step 1 is use a mortar and pestle to break up the food into smaller particles. Okay, so once you've broken it up into smaller particles, you're then going to add this crushed food into a test tube. Add it into a test tube with water, and you're then going to shake it and mix it so that the food kind of dissolves in the water. And then what you're going to do, exactly, grind the food, add distilled water, and you're then going to filter it out, because you don't want the food particles just in the test tube. That'd be disgusting, and it also wouldn't work. So you now want this liquid which contains food in it dissolved. Then we move on to the next part, which is testing. So you want to say, if you're testing for fats, you can use ethanol, because fats don't dissolve in water. So when we're doing mixing the food with water, if you're testing for fats, you're not going to mix it with water. You're going to mix it with ethanol, because the fats don't dissolve in water. Okay, and now for the tests. So for the starch test, it's iodine solution, and if it exists, then um, it will go from dark blue to black. Well, I believe if it has it, it turns into dark blue black. See, I'm still forgetting now. It haunted me that much. The protein test, I'll actually show you guys the answers. The protein test, you've got the burette solution, and that turns violet if there's protein. For the fat test, you can use a Sudan regiment test, and that has a red layer at the top. But I know there's another test that people often use. So it depends on that. And then for the sugar test, you've got your Benedict solution and sugar. You put it into hot water and it should turn orange. Now, don't forget to put it in hot water, okay? A lot of people forget that step and they actually lose a mark because of the annoying mark scheme. So don't forget that. Okay, we're now looking at the next part, which is rate of enzymes. Okay, so the effect of the pH on enzymes required practical. So this is another required practical. Okay, so this is another required practical that you're going to need to know and be able to blurt out. So this is when you have that kind of paint palette looking thing, and you put it's called a spotting tile, and you put a drop of iodine into each spotting tile. You then take 3 test tubes with 2 centimeters cubed of starch, 2 centimeters cubed of amylase, and 2 centimeters cubed of pH 5 buffer solution. Now, unfortunately, if you don't understand enzymes that well, this will just be confusing to you. So make sure that you revise it if you're confused with this. You're then going to put all 3 test tubes in a water bath at 30 degrees. You want them to be control variables to all be at the same starting temperature, but also making it 30 so that obviously it's heated, so it's going to work a bit faster at its optimum condition. Okay, and notice it's at 30 degrees, which is close to the body temperature because obviously it's going to work well in our body. You're then going to transfer 1 drop, but you're going to mix all 3 solutions and transfer 1 drop of the solution into a spotting tile every 30 seconds until the iodine remains orange, until it doesn't change colour. If you're confused as to what I'm saying, when I release the answers, you can hopefully see the mark scheme there. Okay, describe the effect protease would have on baby food. Now here is it's a difficult application question. So with this application question, you need to think, okay, what is baby food? Like, what is the purpose of it? Well, baby food is clearly mushy so that because babies don't really have teeth, especially when they're just really young. So when they need to digest the food, it needs to already be quite small and easy to dissolve. So when we're thinking about protease is an enzyme. Enzymes break down food from proteins to amino acids, and therefore it makes the food softer and runnier. Okay. Describe how ribosomes and mitochondria help to make enzymes. So, this is again a bit of a higher topic question. So, ribosomes synthesize proteins and enzymes using amino acids. Mitochondria releases energy for the process to occur. So, that is just using your knowledge of the different parts of a cell to relate it to the question. And the final one of the page, explain the lock and key theory of enzyme action. I think Nicole already answered that in the chat. Well done for that. That was perfect. So the enzyme has an active site, which is a specific shape. This is then complementary to the substrate, the food that is breaking down. It would then bind onto it. The enzyme then works on the substrate, breaking it down into smaller products. You can call it an enzyme substrate complex, exactly. And then the products are released and the enzyme is unchanged. It's often important to say the part where the enzyme is unchanged because sometimes it can actually take away marks if you don't mention that. Okay. We're now moving on to the next topic, which is the heart, blood, and vessels, which I saw that people were thinking of acronyms for, which is really cool. Okay, so describe the levels of organization in the human circulatory system. Okay, so this is effectively talking about how does the heart work, how does blood get pumped, how does oxygen get around your body. So the thing is, though, they're not just talking about it in terms of how does the circulatory system work, it's saying the levels of organization, which means they want you to talk about going from a cell to a muscle, going from a muscle to cells to tissues, tissues to organs, organs to organ systems, etc. So how does that work? So these 6 markers won't necessarily come up. I'm actually providing the harder 6 markers that students have typically gotten wrong. So this is not paper 2, this is paper 1. Okay, well done, Jude, for the acronym. That looks interesting. Okay, so muscle cells can contract and respire, and respiration occurs here. The muscle cardiac tissue then works together to contract and respire, then producing the heart organ, or helping to work together so that the heart can contract and relax. The muscles make up the valves and the blood vessels. They then put the blood under pressure, and this allows the circulatory system to move to move oxygenated blood and deoxygenated blood. It consists of the heart, the blood vessels working together. Okay. Explain why the left side of the heart is thicker. Well done, Jude. Jude's my favorite student here. Look, the left ventricle pumps oxygen around the entire body, okay? Whereas the right ventricle is only pumping deoxygenated blood to the lungs. So firstly, the blood is actually thinner, the deoxygenated blood, and then there's less pressure. So the left ventricle has higher pressure and the blood is just thicker in general. So the left ventricle needs to generate a greater force of contraction and thicker walls to withstand the pressure. Okay, so that's the bit that people often forget. So in order, because it's not just because of the high pressure blood, but in order for the left ventricle to then pump it, it needs to generate a strong contraction force and therefore put in a lot of energy. Yeah, Wanya, it does mean without oxygen. Okay. Now moving on to the next one, compare the structure of an artery with the structure of a vein. So, arteries have a thick layer of muscles, whereas veins have a thin layer. Arteries have a narrower lumen, whereas veins do not. Veins have a thicker one. And lumen is kind of the gap in between. Veins have valves, which prevent the backflow of blood. Arteries don't because they're already going at such high pressure. Arteries have more elastic tissue as well, so that it can expand and contract greater. Okay, and just for the last question, describe the double circulatory system. So this is simply a 1 marker, so you just want to talk about how deoxygenated blood is pumped to the lungs. It gets oxygenated and it's done via one side of the heart, and it comes back to the body via the other side. Okay, here are the answers if you need any reference to them. And now let's move to the next question or the next topic, which is ventilation and gas exchange. Yeah. Explain how human lungs are adapted for efficient exchanges of gas via diffusion. Okay, so how are lungs adapted for efficient exchange? So here you really want to talk in depth about alveoli and all of the advantages of it. So there's lots of alveoli in the first place, which means there's a large surface area to volume ratio. They have a large network of capillaries, which means that there's a large blood flow so that they can have a rapid transmission. The capillaries are also quite thin as well, so there's a short diffusion path for the oxygen to come in and the carbon dioxide to go out. Because remember, the alveoli is gas exchange, so oxygen is coming in, but at the same time, carbon dioxide is leaving. And you can also talk about an interesting adaptation here. You can say that the breathing mechanism helps as well. As the diaphragm is moving up and down, it moves air in and out. And because of that, this then allows fresh oxygen and the removal of carbon dioxide. Exactly, ventilation. Well done, Jude. That is correct. Ventilation allows for rapid diffusion. Okay, so here is the answer. How many points should you list? Well, it's not a case of listing if there's 6 marks, 6 points, not all the time. Sometimes you just want to say, sometimes you just need to say enough, like you don't need to say 6 points all the time. You just need to have sentences with the points in general. So, for example, where it says breathing mechanism and diaphragm moves air in and out to bring fresh oxygen and remove carbon dioxide, that's a 2-mark statement because you've got the initial bit and you've got the effect of that. Okay, moving on to the next topic. Coronary heart disease. Okay, this is the next one. So, can you guys see the questions? They're quite small. Hopefully you can. If not, I'll read them out. Okay. Yeah, Jude is the reason why you need 80% for a pass. Although I don't think it's going to be 80%. It's going to be a bit lower than that. It can't be that high, can it? We'll see. Let's see how smart your year is, how well prepared they are. Okay. Explain why a stent prevents a heart attack. You're not going to get a whole question on fish gills. You may get a 1 or 2 marker, but it won't be a whole question because you guys haven't specifically learned about that. Okay, so why does a stent prevent a heart attack? How does it work? So you want to say it keeps the coronary heart open. You want to talk about arteries and how that is kept open so that the blood flow can then carry the glucose and oxygen to the heart for respiration. Respiration then allows energy so the body can work and therefore heart attack doesn't happen. Okay. Well done, Santosh. Well done, Wanya. Well done, everybody. Okay. Next question. List the risk factors of coronary heart disease. So often people get confused when they think, what is a risk factor? Well, a risk factor is something that can literally cause it. Okay, it's not something that happens because of the disease. It's something that could cause you to get CHD, coronary heart disease. So you want to say something like lack of exercise, smoking, stress, alcohol consumption, having a bad diet, or a high-fat diet. Obesity is a bit of an interesting one because sometimes they reject that in the mark scheme. So instead, you want to say just diet, sugar. You have to be specific because obesity could be be because of a reason not relating to health, perhaps. Okay. Evaluate the use of cholesterol blockers versus statins for a person with high cholesterol. Now, don't get scared. I haven't actually provided you because this question came with a bit of content telling you about cholesterol blockers because you've learned about statins, but you've not necessarily learned about cholesterol blockers. So you're going to need to see that for that question, but I'll go through them anyway. So statins can have the possibility of damaging organs, but cholesterol blockers do not. Statins could actually cause death, but cholesterol blockers don't. And you would be given this information in the question because you're not supposed to know that. Yeah, you're not supposed to know too much about cholesterol blockers. They will give you that information if you are asked about it. Statins are for lifetime, like as in you have to constantly take it, whereas cholesterol blockers aren't. Exactly. Well done for saying they're not a long-term solution, guys. Okay. And statins could actually have the possibility of reducing cholesterol to 0, but cholesterol blockers will only reduce it. Okay. So statins could completely eradicate it. Exactly. Now moving on to the next one. What condition may be treated using an artificial pacemaker? Now this is interesting. It's a 1 marker, but you have to think a lot. So what is the purpose of a pacemaker? The pacemaker is what helps your heart beat regularly. Okay, so what you then want to say is that what would require an artificial one? It must be an irregular heartbeat. Yeah, you could get stents. So a stent will only be working in one part of your artery. So say if you're because what could happen is if cholesterol blocked one part of the artery, the other part of the artery may still be blocked. So you have to keep putting stents in, but obviously there's a limit. So yes, stents, you need to replace them. Okay. Irregular heartbeat, that is the answer for that next one. Or they actually have a name for it, but I can't exactly pronounce it. It's like carcythmia or something like that. Okay, now moving on to the final question on this page. Compare mechanical valves to biological valves. Okay, well done for everyone answering. So mechanical valves are more durable, they are longer lasting, whereas biological are actually last less time. Mechanical valves can cause blood clots to the brain, which can be very damaging, obviously. Also, with mechanical valves, the patients need to therefore take anti-clotting medicine, and if they forget to take it, that could have a massive impact on their life. Mechanical can transfer infections, yes, but also biological can do that as well. Yep, well done, Stephania. Biological can get rejected because what happens if anything living enters our human system, our body will always attack it. They'll always think that there's something wrong if it enters our bloodstream. So unless it's obviously for fertilization processes. So anything living that's in our system, our body's going to attack. Therefore, if we're going to have a biological valve, our body might actually attack it. So you need to make sure it fits correctly, which could be a matter of chance. With mechanical, there's actually a higher survival rate. Biological, it requires immunosuppressant medication, depending on how much rejecting your body is doing. So here are the answers for those set of questions. And let's now move on to the next one, which is the effect of lifestyle on non-communicable diseases. Yeah, you could argue that there's a shortage of biological valves. Yeah. Okay, explain the risks of a pregnant woman smoking and drinking. Okay, so just think about what are the harms of smoking, what are the harms of drinking, and what that could lead to, and try and use your own knowledge on this. So you can say for smoking, carbon monoxide is produced, which is a toxic gas. And what happens is the carbon monoxide pairs to your hemoglobin in the place of oxygen. So your body thinks the carbon monoxide is actually the body thinks the carbon monoxide is actually oxygen, and therefore you're not actually getting enough oxygen. It can lead to stillbirth and miscarriage, but you need to talk about the steps leading up to that. Yeah, fetal alcohol syndrome, that's perfectly fine. Well done, Nicole. Slower brain development, yep. Could cause malformed babies. So, if there's less oxygen in the red blood cells, this could be really bad and fatal for the fetus, and that could then cause miscarriage, premature baby as well, and fetal alcohol syndrome. Alcohol in the first 3 months can increase the risk of the baby being premature, and if it's heavy drinking, it could even lead to a miscarriage, unfortunately. Okay, we're now moving on to describing the effects of liver failure on the human body. So, this is a 4-marker, but I've actually put a really long answer for this because it's a multi-step thing how the liver links to other parts of the body. So if the liver's not working properly, that means that the amount of bile that's being produced. Thank you, Vendetta. I appreciate that. Okay. So there's less bile produced, so that means that fats and lipids are not fully emulsified. If they're not fully emulsified, then the pH in the small intestine won't be neutralized, the enzymes are not working effectively, food is not getting digested effectively, your person is going to experience severe weight loss. Lactic acid, as people were mentioning, well done. They're not broken down. Accumulation of lactic acid is quite toxic and the body can be poisoned. And then also talking about some paper 2 things about proteins and deamination, but of course you don't need to know that for this paper. Okay. Explain how diabetes can cause the body cells to lose more water. So again, this is a bit of a one where you have to think a little bit. So the blood is more concentrated than the solution within the body cells. Water thus moves out of the cells by osmosis through a partially permeable membrane. So quite a simple answer, but you need to really think about how that comes into play. See, look, everyone's making friends with Jude on YouTube. See, I knew Jude was a good kid. Okay. Moving on to the next topic, which is cancer. See, we're going very fast through this. Oh, wow, we're almost done. So this is the last page of questions for this live stream, and it only took about an hour. Well done. Okay. Perhaps we could have gone through all of the biology topic, but I wanted to leave some content for the night before the exam stream. Okay, let's go into this last page. Well done, guys. You made it through. Let's just finish this up now. So, compare the similarities and differences between benign and malignant tumors. Okay, so you want to say similarities and you want to say a bit of differences. So, similarities, both are uncontrollable growths and both are formed from a person's own cells. That's a point you could say. Differences, benign tumors are actually enclosed, so they're fixed within a membrane so that they can't invade neighboring tissues and they can't form secondary tumors. With malignant tumors, they are not enclosed in a membrane and therefore, well done, guys. Well done, Slinky. Yep, they can invade forming secondary tumors. So invade other parts of the body through the bloodstream. Be careful for saying both are tumorous because the examiner could read that as they can both therefore spread into the body. Okay, moving on to the next one, which is name some risk factors of cancer, so some things that could cause it. Obviously, cancer is spontaneous a lot of the time, although there's risk factors that cause the chance of it to increase. There's nothing that directly we can say will cause cancer for everybody receiving that. So HPV, you could say, carcinogens in smoking, alcohol intake, UV radiation, say if you're going to be going, so UV radiation as in, yeah, lifestyle as well, like sugar diet. Diet has a heavy impact on that. UV radiation as in, what's it called, suntan? When you're in those things and getting a fake tan, yeah, fake tans as well, they can massively impact that. Any sort of radiation, but UV is the most common. So yeah, radiation exposure, well done. Okay, next one. Smoking can cause diseases such as cancer. Explain why this is not a communicable disease. Okay, so, well, communicable means it can spread. Yes, sunbeds, thank you, that's the word. Yeah, okay. Cancers caused by smoking cannot be passed from person to another person. Therefore, the cancer is not communicable, or you could say the cancer is not caused by a pathogen, hence it cannot spread as well. Okay, moving on to the next one. Helicobacter pylori, or acid-tolerant bacteria, which can damage the human mucus lining. Suggest how an infection might result in a stomach ulcer developing. So, moving on. I did not know that there's a cancer in dogs that can spread. That is very sad. Okay, so bacteria are not killed by hydrochloric acid, and so they are damaged by the mucus lining. Exactly, cancer isn't a pathogen. That's one answer there. So bacteria are not killed by hydrochloric acid, and so they are damaged by the mucous lining. Therefore, the hydrochloric acid causes an ulcer because it is directly contacting it. Okay, and our final question of the live stream. Describe how a person infected with Helicobacter pylori could also develop liver cancer. So obviously this was a specific question related to the context of the question. So you want to say, you want to talk about they'll only develop liver cancer if it's malignant. So you want to talk about whether it's malignant or benign and then speak about that. This part is only for triple. I'm not too sure if cancer is in the combined specification or not. Yeah, so you'll have to check that, unfortunately. Sorry, I don't know that. Okay, I'll show you the answers now. If you want to see the live stream timetable, because I have all of the live streams that I'm planning to do in the next couple of weeks, you can find that on my website or go to the community tab and scroll down. Okay. Free science lessons on top. I think no for GCSE on top. Okay, so thank you guys so much for watching. If you found this video helpful or this live stream helpful, feel free to subscribe and share this if you haven't already. Thank you so much for interacting in the chat. You guys have been working really well, and I'll see you in tomorrow's live stream if you'll be there. I cannot remember what tomorrow's live stream is. Let me check. It is RS. Oh yeah, tomorrow's live stream is RS. So definitely come to that. We'll be going through 12 markers, looking at some model answers, etc. Okay, good luck for your exams and